Hi, I'm Morteza Bajgiran. Today I'm going to present my project, which is transcriptomic study of coding genes and long non-coding RNAs during aging in Drosophila melanogaster. So as you know, aging is gradual decline of physical, mental, and biological state of an organism with time. There are many genes and pathways have been studied that underlying aging, and they are categorized into 9 main hallmarks. The primary hallmarks of aging are genomic instability, telomere attrition, epigenetic alteration, and loss of proteostasis. And the secondary hallmarks of aging are deregulated nutrient sensing, mitochondrial dysfunction, and cellular senescence. These hallmarks will cause stem cell exhaustion and, at the end, alter intercellular communication. And long non-coding RNAs are transcripts longer than 200 nucleotides. They cannot translate into proteins. They don't code any protein, and their function mostly is unknown. But a wide range of functions have been discovered for them. For example, they can regulate chromosome architecture. As an example, in X chromosome inactivation, they have a role. Also modulating interchromosomal interactions, regulating mRNA stability, uh, through helping to be scaffold for microRNAs, and also they can do by being a scaffold for other proteins. And also there are some long non-coding RNAs uh, in ribosomes, and they regulate the translation. So there are some studies on the uh, role of long non-coding RNAs in aging. However, mostly they are focusing on some specific hallmarks of aging or age-related disease. For example, senescence-associated long non-coding RNAs, telomere-associated long non-coding RNAs, and long non-coding RNAs that are engaged in cancer or other age-related diseases like Alzheimer, Parkinson, and cardiovascular malfunctions. So, but there are not that much direct study on the role of long non-coding RNAs in aging. So in this study, we are using Drosophila melanogaster as a model organism because they have very short life cycle, so studying aging would be easier. Also, they are easy to culture and maintain, and they are very valuable for molecular studies since 75% of disease-related genes have homologous in flies, and 60% of Drosophila genes are conserved in humans. So the Drosophila can be a suitable model organism for fundamental study, especially aging. So to summarize the, the introduction, I have to say we have many genes and pathways, coding and non-coding genes that are involved in hallmarks of aging. When it comes to long non-coding RNAs, there is still lots of information that we don't know and mostly are unknown. However, long non-coding RNAs comprising more than 12% of annotated genes in Drosophila. So it's an important question to know what group of genes have the most differential expression and how long non-coding RNAs are involved in aging process. And this is our question. So to reach this goal, we have two objectives. First, identification of pathways and group of protein coding genes that have the most significant differential expression during aging. And second, identification of long non-coding RNAs that are involved in different stages of aging. The method I use is that I age the flies for 60 days, up to 60 days. Then I extract the RNA. After that was library preparation and sending for sequencing, and after that uh, analysis of the RNA sequencing data and validation by qPCR. So the reason I use day 60 is that uh, we did we tried to test how long flies can survive. So we saw up to 70 days we can have some flies that survive. However, few flies can survive this much, so we choose day 60 as consider day 60 as old fly, and we compare that with day 1 fly. So uh, after I sent for sequencing, I got the data. The differential analysis that I've done shows that out of 17,808 annotated genes, around 60% of genes, which means 10,738 genes, were differentially expressed in old flies compared to day one flies. And 5,097 genes were upregulated and 5,641 genes were downregulated. After that, I did the enrichment analysis to see which pathways and gene ontologies are more enriched and the more differentially expressed. Based on that, in gene ontology, as you can see, for coding genes in cellular components, you can see they are the most significant gene ontology terms that differentially expressed. However, the one that I made red are in line with previous studies and are related to hallmarks, main hallmarks of aging. As you can see, for example, we see lots of change in genes involved in spliceosome and splicing. And on the other hand, we can see the downregulation in genes related to mitochondria, like mitochondrial inner membrane and mitochondrial respiratory chain complex. In also gene ontologies features biological process, we can see that also again 174 genes related to spliceosome and mRNA splicing upregulated, and also genes related to DNA repair and cellular response to starvation upregulated, which are related to the hallmarks of aging. Beside that, you can see the genes that are green, they are also differentially expressed significantly and they are related to neurological process. And as you can see, neurogenesis shows the most significant and enriched category. And if we study the enriched pathway, you would see the most significant pathway that shows differential expression is a spliceosome, 99 gene change in a spliceosome. And also ubiquitin mediated proteolysis, also 60 gene upregulated. And as you can see, other type of DNA repair like nucleotide excision repair, mismatch repair, homologous recombination, Bex excision repair also upregulated. And oxidative phosphorylation, which is related to the process of mitochondria, downregulated. So when it comes to studying the long non-coding RNAs, after we isolate the long non-coding RNAs that differentially express, we try to see which long non-coding RNAs are close to some coding genes and can have the potential to be cis regulator of those genes. So based on that, we want to see these long non-coding RNAs are in cis position of which genes, and we saw that the most of these genes are the genes that you can see in this slide. And the one is red, also again related to hallmarks of aging, like response to hypoxia and mitochondrial electron transport, which shows that the long non-coding RNAs that related can be the cis regulator of genes related to mitochondria upregulated here. And also many long non-coding RNAs can be potential cis regulator of genes related to neurological process downregulated. On the other hand, also we can see in pathways also long non-coding RNAs that are related to oxidative phosphorylation upregulated, and long non-coding RNAs engage in again oxidative, 8 of them in oxidative phosphorylation downregulated, and also long non-coding RNAs in mismatch repair downregulated. So in general, if I want to summarize, the red one is upregulated and the blue one is downregulated here in this slide. As you can see, genes related to DNA repair, different type of DNA repair, upregulated. Genes related to mitochondria, downregulated. And genes related to cell cycle and cell division, also upregulated, which is in line with previous studies on senescence. And also genes engage in cellular response to starvation and alternative splicing upregulated. So there are some interesting findings also in this study. By studying gene by gene, we can see first of all non-homologous end joining compared to homologous recombination become more active in old flies. There are few studies before that they also mentioned this, but we need further study to see why non-homologous end joining become more active. The problem is non-homologous end joining is error prone, so it will cause more damage, so maybe it can even explain better the genomic instability in aged cells. Also, another thing we saw, the genes related to maturation of Okazaki fragments mostly upregulated. 99% of them upregulated. And the thing is, based on previous study, there is no study on the relation of maturation of Okazaki fragments and aging. But what we know is that if the impaired maturation of Okazaki fragments can cause senescence, cellular senescence, which is one of the hallmarks of aging also. So it would be interesting and need further study also to see how the maturation of what is the effect of aging on maturation of Okazaki fragments. Another thing is that the RNA polymerase 2 has to choose between coding strand and antisense strand for transcription. And there are some factors, mostly epigenetically, they regulate this RNA polymerase to choose one of the strand based on the situation and condition. We saw the genes that are regulator of this process changed in a way in old flies that help the RNA polymerase to choose antisense strand. It means that we would have the pattern of antisense transcription will change as we age, which is also first time that we can see this process in aging. There is no previous study on that. And doing chip sequencing and more sequencing on just antisense transcripts can be beneficial to study further about this. And the last one that we saw also already in other slides, the genes related to spliceosome and splicing process significantly upregulated, and also it shows that most likely the pattern of splicing changed due to aging, and so doing more transcriptomics studies, profiling studies for this also can be interesting, and also it has potential to be another hallmark of aging, and it's first time that we are reporting this. However, there are some studies that shows the role of splicing in change of splicing pattern in cancer, but we don't have any study on the role of splicing in aging. Thank you so much.